So as Frank mentioned, I'm going to tell you a little bit about what we've been doing to develop broad spectrum antivirals, which we hope will have immediate utility and utility in the future for future emerging coronaviruses. And um, my interest in coronavirus started a while ago when I was actually in grad school. So I have a long-standing interest in studying how coronaviruses emerge and to try and develop therapeutics that can address host range expansion and spillover. And this is just an example of that. This is a title slide from a student seminar of mine from 2007, kind of melding my interest in both infectious disease research and rock and roll. Um, and after my time in the Barrick lab as a grad student, I did a postdoc with Charlie Rice at Rockefeller. I actually moved back to North Carolina to work in industry. So I worked at Glaxo for about a year and then came back to UNC as faculty in 2015. And so since the founding of my laboratory in a not so distant past, I've been kind of focused on three areas: building models of viral pathogenesis, where we focus on coronavirus and hepativirus. Also on we're trying to understand how host genetic diversity affects the outcome of these infections. And lastly, kind of using these models to develop broad spectrum therapeutics and vaccines. And so today I'm going to really focus on coronavirus. Everybody knows about coronavirus by now. Even on the right-hand side, there was a drawing by my daughter who was 8 years old. She's now 12. So she had a pretty deep understanding of coronavirus pathogenesis, and she was just a kid. So I think the pandemic has really taught people a lot about epidemiology and infectious disease and virology. So it's been great from that standpoint. But one thing that people might not appreciate is before 2002, the kind of the landscape of human coronavirology was really different than it was today. So what I'm showing you here is a table from my virology textbook from grad school, from the coronavirus chapter, and I've just highlighted the two known human coronaviruses back then, both of which cause the common cold. And most of the viruses that fill out this table are of agriculture and domestic animal importance. And human coronavirology was really turned on its head with the emergence of the original SARS coronavirus in 2002 and the discovery of MERS in 2012. And it really created a new paradigm for human coronavirology that kind of has stayed with us until today. With the spillover of SARS-CoV-2 into humans, it really demonstrates that the coronavirus family has emergence potential, and pretty much all of the 7 known human coronaviruses indicated on this slide are believed to have resulted from spillover from some animal reservoir into people. So, for example, OC43 is very closely related to bovine coronavirus, and through molecular clock analysis, it's believed to have jumped from cows into people sometime in the 1800s. So, because this family has emergence potential, and we've seen 3 novel human coronaviruses emerge in the past 20 years, what are we going to do about it? And what we've been doing is we've been trying to develop broadly active therapies. So we don't want to just develop a MERS-specific drug in this example, taking a one drug, one bug approach, where we make a very specific, very potent antiviral that may not work outside of MERS coronavirus. So we've been taking this one drug, many bugs approach, where we want our therapies to work against the coronaviruses that we know about today, but also those that might emerge in the future. And how we kind of get a feel for this crystal ball prediction is we test our therapies against viruses found in animal reservoirs that may be the source of future emergence. So my specific story that I'm going to tell you about starts in 2015 when I came back to UNC as a research faculty. And it was around the time that there had recently been a pretty significant nosocomial outbreak of MERS coronavirus in South Korea. So MERS, after it was discovered, this is an epi curve just showing the number of MERS cases over time. This virus is endemic in camels in Saudi Arabia and Northeast Africa. And the worry was that someone will acquire MERS, they'll get on an airplane, they'll take MERS with them to another country, and if the virus is efficiently spread from person to person, could result in a local outbreak, epidemic, or pandemic. And we saw kind of the potential for this in this South Korean outbreak of almost, I think, about 200 cases caused by a traveler who had visited Saudi Arabia, gotten close to a camel, and taken MERS back home with them. And so this was on people's minds, and NIH actually invested in some centers for excellence in translational research. Ralph Barrick and Mark Dennison actually were part of one of these centers that got this award. And the one that we were originally part of, and this thing is still around, is headed by Rich Whitley at UAB. And the goal of the project that I was working on was to develop antivirals against MERS and related coronaviruses, given the situation that was happening in the prior slide. So since then, we've kind of been working on a platform for the comprehensive evaluation of antivirals targeting the coronavirus family. And the first kind of part of that platform is built upon virus reconstruction. So genetic systems kind of invented in the Barrick lab allow for the synthetic reconstruction of coronaviruses and the creation of recombinant viruses in the lab. So if someone identifies a new coronavirus in, let's see, in California or in Venezuela, and they publish that sequence, we can make that virus in the lab and study its biology. And it becomes a tool allowing us to kind of measure vaccines and therapeutics against. Secondly, we have in vitro systems based in cell lines and human primary cultures to study these panels of viruses. And then we have models of viral pathogenesis. We pretty much only work in mice, which allow us to assess both prophylactic and therapeutic efficacy and the impact of therapy on not only virologic metrics, but also pathologic and physiologic measures of disease. So the first system I want to talk about today is the primary human airway epithelial cell culture system. This is a model system that recapitulates the conducting airway, the human conducting airway, and both its cellular architecture and complexity. So it has ciliated and non-ciliated epithelial cells that beat in a coordinated fashion. It has mucus secreting cells, so you literally have to wash the snot off of these cultures every few days since they can't sneeze. And early on in the pandemic, Kate Edwards in the Barrick lab infected one of these cultures, fixed the culture, and Camille Eyre in the Marsko Lung Institute took these really amazing EM photos where you see SARS-CoV-2 virus particles just intermingled among the cilia on the apical surface of these cultures. So this is what your conducting airway looks like when you're infected with SARS-CoV-2. So prior to the emergence of SARS-CoV-2, we'd been working with Gilead to assess the potential of a small molecule now called remdesivir or Velcuri and its ability to inhibit coronavirus replication. So an example of that in these primary cultures is shown on this slide. So this is from a 2017 paper where we show a reduction, a dose-dependent reduction in infectious virus production for all these different genetically related yet unrelated coronaviruses, including a common cold causing coronavirus called NL63, a MERS-like bat coronavirus called HKU5, a SARS-like bat coronavirus called HKU3, and then two SARS-like bat coronaviruses that can infect human cells without adaptation. So we say that these are poised for emergence. But the important takeaway is that remdesivir in these cultures was able to reduce replication for all of these different coronaviruses. And data that I'm not showing you is, its impact on SARS-1 replication, MERS replication, and all that. So we and many other people kind of helped create this story here that's kind of laid out in this slide, making remdesivir the first ever direct antiviral approved to treat any human coronavirus. And one thing that this drug had really going for it was that it already been through phase 1 and into phase 2 for Ebola, for another indication. So there was a ton of data on the dosing of this drug and the safety of this drug way before SARS-CoV-2 emergence. We and others at CDC, RML, at NIAID in the non-human primate model, Mark Dennison at Vanderbilt, we kind of played a role in this space in the preclinical development where we demonstrated efficacy against lots of different coronaviruses in vivo and in vitro. And ultimately, it was put into human clinical trial early in the pandemic, and it gained approval by FDA in October 2020. So going from a grad student working on coronavirus and having people say, like, why are you working on coronavirus? Like, you don't have any marketable skills to kind of helping play a role in this story. It's kind of one of the more gratifying aspects of my relatively short virology career so far. Another small molecule that we had preclinical data on prior to the emergence of SARS-CoV-2 has lots of different names. On this slide, it's called beta-D N4-hydroxycytidine. The prodrug of this is molnupiravir, which is a Merck drug that is used to treat people with COVID-19. Prior to the pandemic, Mark Dennison's lab had shown that this drug is antiviral against MHV, a model coronavirus, as well as MERS. And then we and many other people indicated on this side collaborated to do a whole bunch of preclinical work, which helped kind of initiate clinical trials in COVID patients with EIDD 2801 or molnupiravir. And we actually played a role in this phase 2 trial, which is pretty cool, in that we were able to show that molnupiravir therapy in people results in a virus dose-dependent reduction in infectious virus. So we worked out an assay to measure infectious virus in NP swabs from this trial. So basically, we were able to show that this drug exerts an antiviral effect in people with COVID-19, which I think is pretty cool. So molnupiravir, like remdesivir, targets the RNA-dependent RNA polymerase. And this is an enzyme the virus needs to replicate its genome and make its messenger RNAs. And relative to spike, as this neighbor joining tree and heat map indicate, it's highly conserved. So these trees are made from model coronaviruses that span the different genogroups. And the heat map just shows the amino acid identity compared to SARS in the first column or MERS. So if we look at this branch here, which is the sarbecovirus branch, we can see that SARS is 100% identical to itself and maybe about 90% identical to MERS RDRP. If we make that same comparison and look at spike, we can see that there's this precipitous drop in identity when we compare spikes from different kind of genogroups within the family. So if the goal is to develop a broadly acting therapy, picking targets that are highly conserved and that can't change is optimal. And you can really see that in RDRP. So this is a structural model that Kenny Dinnan had made. He was a grad student in the Barrick lab, where he modeled the family-wide genetic diversity of coronavirus on the structure of the RDRP. So conserved residues are shown in purple, variable residues are shown in blue, and the pore from which new viral RNA is generated, as you can see, is all purple, highly conserved. So small molecules that target this region or interact with this region of the protein are likely to be broadly active against the coronavirus family just because of this high structural conservation and sequence conservation among the residues that line that pocket. So I want to talk a little bit about some of the work we've been doing more recently, and it's related to two different initiatives. One of them is READDI Inc., so it's a nonprofit associated with UNC, and the other one is our AViD Center, and that's kind of why I'm on this call today with all the other people in these other AViD centers. And what we've been trying to do with both our AViD Center and with READDI Inc. Is to kind of take small molecules that are broadly active, through the preclinical phase and eventually maybe through phase 1, such that these potential medicines are just sitting ready for a future emergence from one of these virus families which are prone to emerge. And I think the remdesivir and the molnupiravir story that I just told is a prime example of how this strategy can be successful, in that we had done a bunch of preclinical and even some clinical development on these two small molecules, such that when SARS-CoV-2 emerged and we knew that it was a coronavirus, these things were rapidly deployed for testing in people with COVID-19. So I think a similar approach can work for these other virus families that we know are sources of emerging infectious diseases. So the story that I want to tell is from my project in our AViD Center. And it's really a collaboration between myself, Ralph, and Jim Saccatini, who is at Texas A&M. And what Jim had done is done an MPro screen, a biochemical screen, with the library that his personal library he has at Texas A&M. And he identified a bunch of hits that inhibited MPro activity. This is probably, maybe 2021, around there. And what I'm showing you on the right-hand side is the structure of the hit and the structure of the hit within the MPro active site. They then, in collaboration with Case McNamara at Caliber, did a bunch of optimization and SIR where they significantly increased the potency of this hit and ended up with a lead molecule called NZ804. So this all happened basically before we had initiated a collaboration with Jim. And so what we ended up doing over the next year or so is really try and get a feel for the breadth of NZ804, how it operates in primary cultures, and whether or not it had promise as a therapeutic agent in our mouse models of pathogenesis. So the first thing I want to show is just kind of an overview of the in vitro data that we've collected on NZ804 and a demonstration of its broad activity in vitro measured against nirmatrelvir. So all these are just IC50, EC50 curves where NZ804 is in blue and nirmatrelvir is in red, okay? And so the top 4 panels are all sarbecoviruses. So SARS-CoV-2, This is a SARS-like pangolin coronavirus. And then these two SARS-like bat viruses, WIV1 and SHC014. We see NZ804 is highly potent with single-digit nanomolar EC50s against these sarbecoviruses. Interestingly, NZ804 loses potency against MERS-like viruses. And looking at the structure, there's actually one residue in the active site that sterically hinders the binding of the small molecule. And so now Jim's lab is trying to find ways to modify NZ804 to improve its breadth against these MERS-like viruses. There was a decent activity against the common cold causing coronavirus OC43. And importantly, bottom right, we show a nice dose-dependent reduction in infectious virus production in primary cells, where both nirmatrelvir and NZ804 are kind of outpacing remdesivir in some dilutions across this dose response. So the next thing we wanted to look at is whether or not we would have therapeutic efficacy in our SARS-CoV-2 mouse model. So this is mouse-adapted virus in a straight BALB/c mouse. I mean, in this study shown on the left, we have mice dosed with vehicle, mice dosed with NZ804 once a day in red, twice a day in yellow. Nirmatrelvir had a different formulation, so it has its own vehicle, or we're giving nirmatrelvir BID. All these are dosed equivalently at 200 mg per kg for the study. And so on the right-hand side, we're showing body weight loss, where we see we can prevent body weight loss with NZ804 given even once a day, which is kind of cool, or twice a day in yellow as compared to its vehicle. And as has been shown in the past, nirmatrelvir also can prevent body weight loss over time. But importantly, we see significant reductions in virus titer, but measured by plaque assay on the left, where we see even once a day, NZ804 can drop viral loads about maybe by almost a log. And if given twice a day, we see over a log reduction by day 4. So that was really encouraging. And the antigen staining on the right basically just kind of confirms that the quantitative data on the left. So because the coronavirus family is prone to emergence, like I said before, we've seen the emergence of 3 novel human coronaviruses in the past 20 years. We've also seen the emergence of novel swine pathogens during this same time frame. So the issues that we see with the emergence of novel coronaviruses to cause disease is not a uniquely human issue. This is probably happening all the time in both wild and domestic animals. So I think there's a continued need for the development of therapies, which is why I think the work that you all have been doing in these AViD centers is really important for us to be kind of better prepared for future emergence. And another thing that I think is really important to kind of address is that I didn't really fully appreciate until the pandemic happened, but is the ability for these viruses to change in response to its environment. And you can really see that with the evolution of novel variants over time in the human population. And this can have dire consequences to therapeutic efficacy depending on the nature of your therapy. And we saw that over time, forever, with these amazing monoclonals that were developed during the pandemic. And it just took like one change in one variant in the spike RBD to render these things essentially clinically useless. So I think target choice is really important for the future utility of these therapies that we're all developing. And it's likely that we're going to need combinations of multiple antivirals that do different things in the life cycle of the virus to broaden the therapeutic window and maximize the utility now and against future emerging coronaviruses. So I'd just like to thank my funding, shown in the bottom right, our AViDD Center, an R01 that we got from NIAID for the preclinical development of remdesivir, money from the North Carolina State Legislature and FAST grants, everyone in my lab, in the Barrick lab, Mark Dennison at Vanderbilt, and our collaborators in industry and in academia that have kind of played a role in these endeavors. Importantly, Jim Saccatini and Jim's lab for a great collaboration with NZ804.